The overall goal of this procedure is to identify post-translational modifications of proteins involved in plant protein complexes. First, transiently or stably express in planta the tagged protein of interest. Then extract the total proteins from the plant's tissue and immunoprecipitate the protein of interest. Next, separate the immunoprecipitated proteins on an acrylamide gel and extract and digest the proteins from the gel band of interest. Now submit the protein samples for mass spectrometry. Ultimately, the results of mass spectrometry can detect dynamic changes in post-translational modifications such as phosphorylation.
Unlike alternative methods, such as the in vitro incubation of kinase with substrate, this method expresses proteins in planta, so you do not have to know which kinase is responsible for phosphorylation.
This method can help answer key questions in the plant biology field, such as how signals are transducted in response to stress, environmental changes, or pathogen recognition.
In addition to the identification of phosphorylation sites of activated resistance protein complexes, this method can be broadly applied to any post-translational modification of any plant protein complex.
Generally, individuals new to this method will struggle because they are not used to work with a large amount of extract and perform most steps of the protocol in a single day.
For transient expression, grow the Agrobacterium tumefaciens strains to stationary phase. Harvest the agrobacteria by centrifugation at 3000 Gs for 5 minutes. Resuspend the pellets in infiltration buffer. Now infiltrate 4-week-old